Okay, now we're officially moving on. We've come to the point in the open session when it's time for council-initiated discussion.
That's because we thought you were so shy in the last discussion. We're trying to draw you out a little.
So we've all been down this road before. It's your turn to take charge of the agenda. This is a great opportunity for you to let us know about reports that you might want to hear at future council meetings. You are also representatives of the research community. If you know of a problem or issue that's boiling and about to boil over, this is a good time to bring it to our attention. Anything troubling you, anything you would like to bring to NHGRI's attention, we're all ears. Jonathan, start us off.
So we've heard in a number of the concept clearances that the data is going to be going into Anvil. And, I would like to get an update on Anvil. I would like a presentation about Anvil. I think would be really helpful to see where that is. And all these data are supposed to be going there. I'd really like to know the progress and, where it is.
Great idea. Yeah, they're trying to cool us down, Sharon, because it is demonstrably hotter in this room now. So sounds like a freight train, and I apologize.
I have one.
Steve?
I've heard recently a lot of concern from colleagues that are getting feedback about their Chinese collaborations. And if there's been some policy changes and so on, I think it'd be good for us to just hear them.
I think we'd want to invite someone to come talk about that.
Yeah. I can guarantee you. There are people that could come and potentially talk about that. Not me. One thing, there's been, and I announced some things here. Probably the last council I announced it, even the one before. There's been a bunch of institute leadership changes over the last year. We haven't been able to slot people in. We were trying to get Ned Sharpless here, and of course, then he gets pulled over to the FDA. Are there particular institutes or any NIH, people from other institutes, new leaders, or that you'd be interested in hearing from? We don't routinely just bring any new institute director, but some are more relevant than others.
The new data officer?
We could bring Susan, okay?
So we had a little bit of discussion about this earlier, but I've become increasingly curious about how much of NHGRI's expenditure is associated with a top-down acronym designated program and how much of it is investigator-initiated and bottom-up. And I don't know, how often do you present the whole overview of?
So Terry gave us.
We feel like we do that every time now, but if it's not, if that number's not coming across, we could look a different lens to try to give you that number. 49%. 49%? Unsolicited, 49%.
And can that be broken down even further, or is that all investigator-initiated R01s, or?
So that's where it gets complicated. You want to come to them? Our two budget presenters are arm wrestling.
This is always a source of confusion. It's a source of confusion to us. So solicited is anything that responds to an RFA. Unsolicited is anything that responds to a PA. But it can be R01s, it can be U01s, it can be R24, U24s, lots of different mechanisms. If you could help us understand what your question is. I think just a list of mechanisms isn't going to tell you necessarily what you want to know. Perhaps we could talk afterward. We could look over.
For many of the programs that we talked about today for concept clearance, there was the opportunity for R01s to be submitted, but to me that's a little bit different from somebody waking up and saying, I dreamt up something wonderful last night and I'm going to pitch it. I want to know how that type of person initiative fares in in the overall scheme of NHGRI.
Yeah, and it varies by area. So the newer areas, you might imagine, fare worse than areas that are well established. The R01 is a mechanism that can respond to lots of different things, and so that's, you know, anything that we stimulate. Unfortunately, NIH now, or fortunately, has an approach that everything has to respond to some program announcement. So it's very hard. You know, there's what we call the parent program announcement, which is just genomics, send us your ideas. And then there are more targeted areas that we work with you to identify that we want to stimulate. And so, you know, it's probably somewhere in between. But the just waking up in the morning is probably the parent R01, I would think, is where it should go.
I'm over here. Yeah, and so I think, and I don't want to overextend this discussion right now. And so Terry and I are actually going to be talking in closed session a little bit about a topic very related to this. And so we have some preliminary data that we can share with you then. I just don't want to go on record with all the cameras on with something that is slightly preliminary. But as part of that, I think it would be useful for us to get a definition, because we get this feedback kind of often, and I think we're struggling, as Terry said, in getting an understanding of what is it that you guys are wanting, not you as a representative of the community, are wanting to see and understand when we do that. And so when we come to that discussion in closed session this time around, we can help with that, and that can help us formulate an appropriate, well-researched response that we would then be able to do in an open session or other setting. Does that, yeah.
As long as we're talking about resource allocation, I want to bring up something that Steve brought up earlier in this session, which is how much of the budget is devoted to technology development. That's so foundational. I think I agree with Steve. I think it's probably not as much as it should be, and I think that'd be a good thing to discuss.
So what exactly would you like us to present? Do you want just a more granular report of what's in our portfolio that falls under that title of tech dev?
Yeah.
Okay.
And how it relates to the whole budget, you know.
Okay.
And maybe what our goals are. You know, where should it be? Where is it, historically, where has it been, and where should it be?
Faster, cheaper, better quality. Jonathan.
So, as you know, NIGMS has these Mira grants, which provide relatively long-term and relatively flexible funding for investigators. And so, I know that we've done a little bit of this with the program for collaborative scientists, but I wanted to raise the question about whether NHGRI would be potentially interested in putting more into a program like that for investigators more broadly. Like, I think that there's potentially a lot of advantages to investigators, so they can, if they're successful in getting that kind of funding, then it potentially reduces the amount of time that they spend on grant writing, gives their labs much more funding stability as well as flexibility to change directions and things like this. And I think that for many of us who are not substantially funded by NIGMS, like that, we have some people have some jealousy of that that set of people, and I think it's something that NHGRI should potentially consider.
I'm not familiar with that program. Does GM put any boundaries on it? It can be in any area of science that's in their remit.
So as I understand it, it's restricted to people who have funding from NIGMS, and then conditional on that constraint, then they can propose what they want to, and then it goes through a review process.
Go ahead.
They trade the R35 for the R01.
I see. Okay.
Yeah, so it blocks them from getting additional NIGMS funding, I think.
So just as a reminder, and Jonathan knows this, so our Genomic Innovator Award is us sticking our toe, or we gave 6 awards, so maybe we went up to our ankle into this water of trying out this experiment of funding investigators more than the specific projects. And so we feel like we're now in the mix of a number of institutes who are trying this new approach. One thing we might want to consider to stimulate the discussion, is there are, and I don't know how many there are, but I know the folks at GMS are probably keeping track of this because they're sort of been intellectually leading in this, is whether anybody started to look more critically at some of the experience with the programs, which ones there are, which the institutes, what the parameters are, everybody's doing it a little bit different, and whether there's any early lessons learned. And if somebody has done that, maybe they could come make a presentation, and maybe that would help stimulate a discussion and get NHRI thinking, do we want to get down to our knees and stick in the, in experiencing this with maybe another similar program in addition to the genomic? I think we're open to this, but I would just want to be stimulated by the experience being gained by other institutes to help that discussion.
So my observation from the investigator side is that people seem very happy with the program, for what that's worth.
Is that universally true? Because I thought
Absolutely not. I didn't have one of those grants, but some of my faculty have, and it's the way I see it, at least the early implementation of it at GMS, is it's a way to restrict funding. They turned the R35s, they're supposed to be the equivalent of 2 R01s, and I don't think any of them have gone to that level. And people who have converted their R01s into R35s have ended up with less money.
Yeah, I mean, I was in the first pool of people that got them, and I had 2 R01s there and consolidated them to an R35, and it was 20% less than the total of my 2 R01s. And so it's nice that you have 5 years of funding, and they're coming up for renewal soon, and we have no idea what the renewal procedure is going to be whatsoever. So, but I think it is good to have longer term you have the flexibility of there's no aim, so you can do whatever you want. So that part is very nice and liberating.
Yeah, I mean, I think it's probably an interesting point to, oh, sorry. No, you're fine. I think it's an interesting point to discuss because a lot of our grants actually involve, NIGMS grants on average, my understanding, are smaller as well. And it's generally somebody working in their lab. It's not somebody saying, I want to sequence 50,000 individuals, right? So part of the structure depends on whether that's going to work well for your, so it would be a good topic of conversation to look at some of these different awards and which ones would be most efficacious. I do think thinking about the length of the grants just in general is something we should, that I brought up very early this morning, is something to think about as well, whether 4 years versus 5 years and things like that.
Lisa, I'm dying to know.
Oh, I was just going to make some clarifying comments about the R35 in general. So yeah, we are using it right now for the Genomic Innovator Award. NIGMS has implemented it one way. I was on the working group at NIEHS when we started to implement it. I mean, I think it has some caveats, which you've mentioned. It has a cap, a budget cap. It does provide that flexibility. It does require you to commit a certain amount of effort to the project, which limits what you can do elsewhere. So I think that there are a lot of interesting things that we could do with it here at NHGRI, and we need to figure out how it fits in best with our program.
Other thoughts, other requests? Gail.
So this isn't for something for you to cover after I'm gone. This is a question about.
Just to be clear, that's after you've left council.
After I've left.
Not really gone.
This is not the end.
Ho, ho. No, this is not the end. No. So, but somebody suggested to me when we were on the break, and I think it's a really interesting thought, that there's a time-limited issue with and a desire to move forward really quickly to develop the methods that are so challenging and that somebody in the room has published on, Jonathan, and that there are others that at your disposal, perhaps at NHGRI, are other mechanisms that you could use and maybe collaborating with other institutes, NHLBI and NCI, for example, who have these big cohorts, to have a workshop on methods, to convene something that would bring together, really key players in this area and start work on something which everybody thinks is so important. And I don't know if that's possible, but I just thought I'd I'd mention it because it seems, I think voting, well, I just think it would be awesome if you guys could make that happen sooner than somebody write a grant, get it reviewed and get funded and start.
You're speaking of a workshop only, not providing funding. Okay.
Well, I mean, workshops need to be funded. I'm just saying that there's other mechanisms that you guys have at your disposal likely that would be a quicker way to address some of these really important methods questions.
Again, you're talking about a scientific meeting, not supporting research to develop the
Right,
methods.
absolutely,
Okay.
a scientific
Okay.
meeting, yep.
Well, I mean, just to follow up on that, as an example, quite interestingly, when I think Wendy and I I was talking with someone about this morning, the American Society of Human Genetics announced they were going to have a webinar on polygenic risk scores. And, webinars are not normally that they had like 900 people sign up. Now, and I think something like 500 or something got on it. So I think it just shows in the community how much interest there is in this topic. And so I don't think a workshop's going to create methods, but it does highlight that whether there might be utility in having a workshop related to having people that are developing methods come and present and talk, perhaps to help with planning of future grants and things like that.
This is not a new idea, just a comment to connect two discussion points. I mean, to me, when Steve says technology development, of course, in this room, that means hardware and chemistry, but it's also software, just to point out. And, so really, I know that, again, in this audience, you probably don't think about technology as bioinformatic technology, but when you're talking about methods development, you're talking about technology.
This side of the room is too quiet. Okay, well, there's plenty of food for thought here, so thank you very much for that. All right, and you know where to find us at breaks if more things come to your mind.